Good morning, good afternoon, good evening, wherever you are. Welcome to the QTurn 2020 workshop. This is the beginning of the second QTurn workshop on quantum information, quantum computation, quantum foundations. You should be hearing me now. If you don't hear somebody presenting, your mic or your sound is not working, check it out. Hello, wherever you are. My name is Juani Bermejo-Vega. I'm one of the organizers, steerers of the conference, and I want to give you a brief introduction to the event and what we're doing here. So, as you have seen, this year we have the second edition of the QTurn conference, and this year it's happening online because of the COVID pandemic. Initially, the QTurn workshop was planned to be held in India, but it couldn't happen, and we decided to take the opportunity to also move the event online, make it fully international and delocalized geographically. So we have 3 time zones this year. Okay. So before we start, let me say some motivational words. Why are we here? Why are we doing this conference? So we believe, many of us believe that science has to be for society, because science is made by people. Also, science only works if we cooperate, if communities cooperate, if groups of scientists cooperate. And a concern that we had before we started organizing the QTurn conference is that, well, science sometimes can exclude certain people from certain backgrounds, and a model of science that excludes certain types of people can not be very good for society, or can also fail at doing its own job if it's not cooperative. So one of the motivations that we had before starting QTurn was, can we try to make an event, a type of conference where we can build a model of doing quantum information science that's more cooperative. So the main question we have before we started this event is, can we change the format of the conference a little bit and make a type of conference where we also do not talk only about science, but also about the scientists. That means certain kinds of community issues. So that was the next question we had, like, how can we build inclusive scientific communities in our field or in quantum information or in general? The goals that we had before starting the event were to create a space for research, but also for researchers, to create a space where researchers from underrepresented backgrounds could work and share their work and thrive, but also any kind of person, and to create a space where we can talk about community issues of quantum information scientists and researchers. So this is the format that we have. This is what we planned. So the QTurn conference, now happening online for the whole week, is a standard workshop in a sense on quantum information computation and foundations, in the sense that we have regular quantum science program with invited talks, contributed talks, and posters. Many of you are presenting here today, and this is sort of the usual format in a quantum information conference. But we also have something which is different from other events, and that's an awareness program. And this is like a track, a series of talks, discussions, panels, where we try to discuss about community issues, issues of diversity, issues of inclusion, topics of privilege, implicit bias, health, mental health, human rights, worker rights. Also, we discuss safety and ethics, but generally whatever that relates to scientists, community issues of scientists is discussed here. We will have 6 talks on this. We also try to make the event inclusive to any kind of person. So for this reason, we try to foster a safe space. We offer a safe space for everybody. We want everyone to feel welcome and safe, in particular, people who belong to underrepresented groups in science, technology, engineering, and mathematics. This includes women, people of color, LGBTQ people, and academics with chronic conditions. This is our contact. You can find us in all social media, and this is we have a mailing list, and we have a website. This is in a snapshot, the Qt 20 and 20 event. We have a quantum science program this year with more than 80 talks. 6 of these talks are invited talks, 79 contributed talks, and we have 77 posters. We also have 6 awareness sessions, and we had a huge amount of registrations this year. This is 902 registrations. In the first Qt that we organized, we had about 113 registrations, so we have grown a lot. And this is also probably because of the fact that we are doing the event online. But it's a great success, and we are very happy to have you all here. This is how the event is going to work. We are going to have 4 software tools, and please pay attention now. So this is very important for attending the talks and posters. So the first tool we have is Slido. Slido is a tool that we ask you to use to attend the talks and ask questions. And we also have Zoom, which is for speakers and chairs to present talks and discussions. Very important. If you are not a speaker or a chair, you should join the talks through Slido. And if you are presenting or participating actively, we ask you to join via Zoom. Slido is much better for us for asking questions and moderating the questions. And you also have a voting system where you can vote your favorite questions there. So ideally, join via Slido, unless you are presenting. Also, if you have a strong need for seeing live captions, live subtitles, maybe you don't see the slides very well, then you should join Zoom, because in Zoom there is a live captioning feature. We also have another software which is called Remo, and Remo will be used for poster sessions and organizing social. It's sort of like a video game with conference tables where you can socialize online, and it's very nice. And we also have Slack. Slack is the central hub of the conference. It's where all links are, where everything you need is collected, and where it's also a place where you can interact with participants, put information about your posters, and read about all of these things that we're explaining now. So the central hub, Slack. If you go here and you feel lost, your main friend in Slack is going to be Zero Mainboard, this channel here on the left-hand side. On this channel, Zero Mainboard, you have a summary of all information that you need to navigate the online event. So we have lots of tools and lots of features to make the event dynamic, and they are all summarized here. It's very important that you read the welcome guide. The welcome guide explains you how to set up your profile. Please read the welcome guide, and it also explains you all guidelines that we have for making your presentations and pleasant, and it explains you how to set up your talk and posters, and it explains you literally everything. So this is your main place. Of course, if you have questions or you have technical problems, we have a channel for this, and you should go then to #1technicalsupport, and we have a fantastic team of technical people there who will help you if you have any issue. Please check also the timetable, the code of conduct, the privacy policy, and the chair and speaker guide. This is the team behind the event. We have a very motivated, fantastic, great team of main organizers: Elisa Agudelo, Ewan Allen, Glaucia Murta, Luciana Henaut, Ravi Kunschfeld, Ralph Silva, and Sabine Folman. And we also have 3 people in the steering group who are also the founders of the conference, who are Ana Belén Sainz, myself, Juani Bermejo-Vega, and Jelena Gurianova. We also thank the work of our program committee chairs, Magdalena, Mehul, Nana, and Prabha. And they have done a fantastic work of coordinating a program committee of 53 people this year who have done a lot of work for the event. We really thank the work they have done here. The program committee of Qt is usual in a sense to other program committees of other conferences, although we do try to make it more inclusive and diverse. And we also share some guidelines on how to review before the reviewing process happens. These guidelines inform you about issues of diversity and inclusion, like implicit bias. As I said, we have a code of conduct. We want the event to be safe. We want all you to feel welcome. We don't want you to have bad experiences. And for this reason, we are dedicated to providing an harassment-free space for all of you. And please read the code of conduct. If you happen to have an unpleasant situation during the workshop and you think this violates the code of conduct, you should reach out to the organizers, and you can do so by going to qtern.org/code of conduct. There you will have some information on how to report harassment during the event. There are two forms of reports. You can use a form that can be made anonymous or non-anonymous, and you can also contact organizers directly there. It is better that you use the form if you want a fast reaction, and if you want a more, I don't know, if you want to contact somebody directly because you feel more comfortable doing so, well, it might take a bit longer, but you can also do it. So, of course, this event wouldn't have happened if we had funding support. So for this reason, we thank our sponsors, FQXI, the Matter and Alike Institute for Quantum Computing, MLQ4, the International Center for Theory of Quantum Technologies, the University of Dansk, Microsoft Quantum, Cambridge Quantum Computing, and River Lane. We thank an individual donation by Chris Garnett, Dr. Chris Garnett, and you can read more about our sponsors at our sponsor webpage in our webpage. And they have some information there about what they do and their efforts in quantum information. And they also have tables at the remote sessions where the socials will happen. We also like to briefly thank other people. This year it has been a very difficult event to organize because of all the changes from a physical event format to a virtual event format. A lot of people help us through the way. We would like to thank Arti Sundaram, Chandru, Genia, Leah, Nitika, Subhare, and Vislasini. And we also would like to thank the people, the technical team behind the event, Delania Kea, Management and Communication. This is a company based in Granada. They are responsible for making everything you see in Slack working. So thanks to all of these people. And with this, we would like to wish you a pleasant event. You can follow us in social media. We are in Twitter, Instagram, LinkedIn, Facebook. This is our email, qtern2020@gmail.com. We have a YouTube channel. The YouTube channel there, the talks will be posted after the event. I forgot to say that during the event, after each talk, we will hang temporary Zoom link in Slack that you can use to see the talks during the workshop in case you miss some talk. If you want to tweet during the event, if you're on Twitter or you are on a different social media, you can use the hashtag qtern20. You can say hi to us on Twitter. We will also put you on a Twitter list that we are doing for the event. And with this, I would like to wish you to have a great scientific and inclusive event.
So thank you, everyone. So let me begin by just thanking the organizers for giving me the opportunity to chair this session, which has many of my good friends in it. In fact, so I'm looking forward to the session, and the conference looks fantastic. So today's session is going to have 6 talks. So there's one invited talk and 5 contributor talks. And the invited talk is 40 minutes long, and all of the contributor talks are 20 minutes. And there's a coffee break in the middle, which simply means that we'll walk away and have a 20-minute break there. And then there is a 40-minute interaction session on Remo at the end of the session. So I'll remind you again towards the end of it. So remember that the session at the end of it is a meet and greet session. So you can speak to the speakers and other attendees. So that's one way for you to have a little bit of social interaction. And I just want to just say that all questions should be directed through Slido, and if you feel the need to ask a question, then please go ahead and post it on Slido. And also feel free to upvote the questions that you see that you would like answered, because confusions, when they arise in for more than one person typically. So if you feel like that question is the one that you'd see. So what I will do is I will keep an eye on Slido. It's being moderated from the organizer side. And I'll keep an eye on Slido. So there'll be a delay between when you post the question and when it appears on Slido. So just be patient. It's just being moderated by somebody. And once the question appears and the upvoted questions appear in front of me, I'll make sure to draw the attention to the speaker to it if the speaker hasn't already looked at it. Right. So if there are any technical issues, as Juani just eloquently put it, please contact the technical team via Slack technical support channel. So there is if you go on your main Slack channel, there's a technical support, and so you can just post there, and somebody I'm sure will just get in touch with you. Okay, so with that, let me just briefly introduce our first speaker. So we have an invited speaker today in front of us, Kasturi Saha. Kasturi is a good friend and colleague here at IIT Bombay. She's an assistant professor with the Department of Electrical Engineering, and she works in the field of experimental quantum optics. So take it away, Kasturi.
Hello, good morning, good afternoon, and good evening in all time zones. And thank you very much to the organizers for inviting me for this, giving this invited talk and sharing our new results on building a quantum diamond microscope. Let me get started here. So I'm Kasturi Saha. For those of you who don't know me, I am an assistant professor in the Department of Electrical Engineering at IIT Bombay. And those of you who don't know where IIT Bombay is, it is in Bombay. It's the Indian Institute of Technology, Bombay, in Bombay, and in India, which is in the western part of the country. Today I will be telling you about our recent efforts in building this quantum diamond microscope. So what indeed is a quantum diamond microscope? So what you are seeing here, and let me just use a pointer. I hope I'm audible clearly, right, Sai?
Yes.
It's fine?
Yes.
Yeah. Okay. And the pointer is visible, right? Yeah. Okay, so this quantum diamond microscope, as you can see here, is a very cartoon picture that I have sort of put together. This is the diamond slab that is, that is our real sensor. And then you can have any sample that you would like to measure, whether it's a magnetic field or stress or electric field, etc. I have just highlighted magnetic field here because most of my talk is essentially going to be focused on measurement of magnetic fields. But it could be anything like electric field, stress, etc. So here what you can see is a sample of diamond. You have some unknown sample that you would like to probe. What we do is we start off with a green laser and we excite it, collect the fluorescence, and in this red fluorescence essentially lies all the information about this magnetic field that we want to sense. And the talk is going to be about telling you how to build and put together this kind of a sensor and also what it has been applied for, etc. So here are some examples from the recent past about some of the stark measurements made with this kind of a sensor, which is a microscope, which is used to image any kind of sample. So what you see here is an example of a hard disk, which is just your normal standard hard disk. And what you see here is the map, the magnetic map, the contrast showing the the magnetic structure within this hard disk. So here in this case, of course, the sample was not the sample is just your hard disk, but the NV center, and we will talk about the NV very soon, is in the form of like an AFM tip, and you have a tip that sort of scans over the sample, and you can profile the magnetic field using this NV center. Now, in contrast, you can have a very, very different application where you measure the magnetic field emanated from a neuronal signal. And that is really going to be the highlight of my talk here. We will be focusing on measurement of neuronal signals and application of the quantum diamond microscope to measurement of neuronal signals. But this picture essentially shows you the first measurement of such, where you have this worm which is called C. elegans, and it has only one neuron which is going all the way from the head to the tail. And as the signal is traveling, it can be thought of like a current carrying wire. A current carrying wire has a magnetic field. This magnetic field has been measured by this quantum diamond microscope. So that's one other example. The next example, again, is another very contrasting example where we can have samples from geomagnetic fields, and this sort of allows you to look at a lot of geomagnetic fields from, say, particles from leftover that has come to the earth or collected from the moon, etc. So these kind of samples are possible, and you can look at, so this sort of a color map shows the variation of the magnetic field as you put the sample on top of this quantum microscope. Right. So what are the features of this quantum microscope so far that have been sort of reported in literature? I'm really focusing on the magnetic field here. So the DC sensitivity is about 50 picotesla per square root hertz. That was reported in 2018. In fact, there has been a very, very recent development on this, and it has been pushed down to even maybe a few picotesla for DC sensitivity, and really pushing the limits and boundaries of measurement of magnetic field with this kind of a quantum microscope. And then you also have this AC sort of a sensitivity, which is 0.9 picotesla per square root hertz, which was measured by Wolf, etc. So whenever we are talking about a microscope, obviously we want to know what the spatial resolution is. And the spatial resolution here, we are diffraction limited because we are measuring with green light. And so it's about 250 nanometers. And then, of course, you can use, optically enhanced techniques, and that allows you to push down to about 5 nanometers in the resolution. But what is extremely nice about this microscope is that unlike any kind of squid or such such sort of tools, comparable tools, where you can have sensitivities really in the femtotesla region, but here you don't really get this kind of a field of view, which is in this case is about 400 micron by 400 micron. Now that's incredible, because you can really zoom into if you can really use these optically enhanced techniques, you can go to about 5 nanometer resolution, but you can have a field of view of about 400 micron by 400 micron. Now that's quite incredible in terms of looking at magnetic maps over a wide area. Now, there have been some very interesting applications of this in the condensed matter community, where people have looked into formation of vortices and image vortices at low temperature using a quantum Lyman microscope. So here you see, if you can follow my pointer, you can see a contrast map of vortices in this. This is a field of view of 20 micron by 10 micron, and you can see the spots, the yellow spots are where the vortices are. And then this is indeed another example which is of a similar kind, where you can see the bright blue spots are essentially where the vortices are formed in this kind of a sample here, which is I think this is a niobium sample. Where indeed what you can also do is you can form sort of contacts and also investigate electron-electron interactions in a wide field of view, and also look at it in a very close form also. So similarly, one can see that you can image the current flow in graphene, and also look at what happens when you have a strain sample, stress sample, and you can see that there is a very nice image of the current flow. And what this microscope really allows you is that it gives you vector information about the current flow, which is which you will not get in a typical kind of a squid measurement. Right. So here is a quantitative number for the current flow, which really enhances the, really shows the capabilities of the tool. And again, there have been some, extensions of this kind of work where we can map, dynamic magnetic properties, etc. Now, these are some of the condensed matter examples, but what I will focus on is really the medical imaging aspect of these kind of NV centers. What I'm showing you here is a picture taken in 1895, must be very familiar for all of you. This is the first X-ray image by Röntgen, and this is Röntgen's wife's hand. You can see that the information that you get out of this picture is essentially the fact that this is just Röntgen's wife hand, and you can see the bones, right? The bone structure. And X-ray to date is still used and is very much in use and will be used in future as well. But the information we get out of it is just the bone kind of structure. Now, as we go to the next kind of more elaborate medical imaging, where we can see that there is some more details of it, but this is still the skull and the, you know, the bones, etc. But as you go use, you know, MRI, where here you can see essentially the sort of a tissue structure that we are able to extract. So we are able to extract out more information from what, you know, Ronjen had, you know, sort of used the X-ray imaging for. So imaging therefore has gone from, you know, just the bone kind of structure, elucidation about the bone structure, to getting information about the tissue structure. And MRI, of course, today is very much in use, and this was developed in 1971, has progressed greatly in the past maybe 20, 30 years, and it allows us to really capture out what is happening at the tissue level, and you can really get a lot of detailed information from this kind of image, right? But the question now is, what I have shown you here is sort of the tissue image of a brain, but now if I want to go beyond that and really understand at the cellular level how these cells are actually interacting with each other, the question that I would like to answer is sort of, you know, can I understand what somebody is thinking? If I can interpret the exchange of information between these two cells, will I be able to interpret what is going on in our brain, what is really happening at the cellular level, and is affected by the stimuli that is given to these cells, right? So what is the correlation between the stimuli and what is happening as the cells interact with each other? So this is the kind of question that we would like to see and understand in the long term. But as soon as I go from this image to this image, the complexities of going from this image to this image is, you know, extremely large. What you can see in terms of numbers is that given this kind of a brain tissue, a voxel size of 1 millimeter cube has about 50,000 cells. And there are about 6,000 synapses, and synapses are nothing but these neuronal. So this is an image of a neuronal cell here. So this is a neuron body, which is the axon, and the soma, and here is the axon sort of interacting, and the interaction is sort of called the synapse. There are about 6,000 synapses in this 1 millimeter cube. There are about 300 million connections in each of. So obviously, as soon as we try to go scale from this image to this image, really our problems sort of exponentially increase, right? And that really is the highlight of the problem that we are trying to address. And the first question that we ask is, can we even map the individual interactions? Can we actually look at what is happening at the magnetic map, in the magnetic maps, or as the current travels down this little wire, or the, you know, the axon connecting this one cell body to another, right? So how is the magnetic field really changing? Can we pinpoint where the signal is coming from, and how do we get to that? So, and that's where we start to understand, you know, and ask ourselves, can this quantum diamond microscope really help us? So therefore, what my talk is going to be today is discussing with you about these color defects in diamond, which is essentially the quantum diamond microscope, tell you about how to measure the magnetic field, the method, and the great challenges and the outlook. And finally, what I will tell you about is sort of looking at the problem from the other side, where we want to understand the neuronal signals and try to reconstruct the neuronal signals and see whether we will be able to indeed measure and identify these kind of interactions between the two cells, right? So that's how the flow of my talk is going to be. So let's jump start. My talk is going to be focused on color defects in centers in diamonds. Now, many of you and probably all of you have seen color defects and in some form, which is essentially in the form of colored diamonds. And these are commonly seen in gem stores. So you would see very colorful yellow diamonds, orange, blue diamonds, and the very famous sort of pink diamond, etc. Right? And of all of these sort of impurities that I mentioned here, which is boron, nitrogen, hydrogen, etc., what you see, what is important for us is the yellow diamond, which is essentially filled with nitrogen. Okay, so typically what happens is when in nature, when the diamond is growing inside the crust of the earth, as diamond, which is essentially nothing but carbon, some of the nitrogen from the air sort of creep inside this lattice of diamond, and then it forms defects in the crystal, and that allows this diamond, which is typically white and very nice and shiny, to become like a yellow diamond, because it sort of emanates in the yellow, right? So here what you see is unlike the gemstone diamonds, we have in our lab a yellow diamond, which just looks like a glass slab. This is just glass, and this is the diamond, which is filled with nitrogen. Okay, and that is sort of just looks like a tiny piece of material, like a sample. And if you do sort of PL, which is nothing but photoluminescence spectroscopy, what we see is a lot of different kind of spectra here. And what is interesting to us are these two peaks, which is the one at 637 nanometer and the other one which is around 560-ish nanometer. Now, these are unlike just the nitrogen. If you have a vacancy next to it, then it forms this something called NV, and that is what is going to be of interest to us. But as you can see, there is a lot of variation that you can get in any sort of, these are different samples that are naturally obtained samples, and you can see a lot of variations in their spectra. Now, what we want when you want to build a sensor is not something that has a lot of variations. So therefore, the sample is of critical importance to us. And what you want are samples that look somewhat like this, which are grown under extreme care, where you have and control, where you want your NVs to be in a very specific location with some specific properties, etc. Right? So unlike the yellow diamonds that I was telling you about, the diamonds of interest to are actually the pink diamonds, where the pink comes from the fact that this nitrogen, when there is a vacancy next to that nitrogen and it captures an additional electron from the carbon lattice, then it forms this NV minus complex. So typically there are three charged states of nitrogen vacancies. So it's NV0, NV minus, and NV plus. And of the three, NV minus is the one that is of interest to us, because that is the one that actually has the properties of a quantum sensor. Of course, NV0 have been recently shown to have some properties of a quantum sensor, but NV minus is the most prominent one. And the most interesting thing is that this sensor actually works at room temperature. This is a room temperature magnetic field sensor. So if you shine green light, if you collect red light, what you see here is an image where you have NV centers with the color bar with a high contrast as the red, and the background is blue here. So the NV which emits fluorescence, the higher the more red it is, the more fluorescence that we are collecting. This is a confocal image, where this is a very, very special sample where we have actually implanted nitrogen to create these nitrogen vacancies. And you can see this is an array that we have created here in a typical sample like this, which is, and if you just look into a 15 micron by 15 micron, this was a specially created sample where you can see the fluorescence coming up. Okay, so this is what you would see if you had a few NVs implanted in this diamond. But typically when you want to work with a sensor, you essentially want many sensors, because your sensitivity actually scales with the number of sensors. And therefore, instead of having an array, if you just have NV centers implanted all over the sample, that is what would be the ideal case, and that is what we would like to use. So these pink ones, which have more number of NV minus, are essentially the ones that we want. We want to shine green light, we want to collect the red light, and that has all the information. What is also nice is that you can work in another extreme where you can actually pinpoint into a single NV center. It's optically addressable, and that can also work like a single qubit. So that has a different application for quantum computing altogether. But we are not talking about quantum computing today. We are going to talk about quantum sensing. And these many such NV centers can form a quantum sensor. Okay, so here is the electronic level structure of the NV minus in this inside the diamond matrix. So within the band gap of diamond sits the NV minus electronic structure. This is the ground state. The ground state is a triplet. It has a 0 and a degenerate plus minus 1 with a zero field splitting of 2.87 gigahertz. If you apply a magnetic field which is essentially you are doing Zeeman splitting, then it just sort of splits out the plus and the minus 1 level. And that allows us to create this sort of a pseudo 2 level system by probing either the 0 minus 1 or the 0 plus 1 sort of a state. Now, how do we really probe the system? We shine green light, we let the we see that the electrons can go either to the metastable state, which is sort of highlighted in the dashed arrow here, or it can fluoresce in the with a coherent system with this 634 or 637 nanometer light. So the beauty of this system is that it can actually polarize, optically polarize to the ground state. If you just keep shining green light, it turns out that the probabilities are such that it will always optically polarize to the ground state. And then you can apply somewhat of a microwave here, and that will allow you to sort of control the state between 0 and plus 1 or 0 and minus 1. And what is again interesting about this sort of a system is that if the NV center were to start from plus 1 or minus 1, it fluoresces 30% less than if it were to start from 0. And that actually gives us a contrast difference between the plus and the minus 1, essentially meaning that you encode the spin information in the optical information. And that is really the power of this kind of a system, where you can actually probe and control the ESR effective qubit with optics. And typically when you have an ensemble of NV centers at room temperature, the T2 star is about 1 microseconds for a very dense ensemble. For single NVs, it can go really up to hundreds of microseconds and even up to milliseconds at room temperature. Here what you see is, sorry, somehow sharing was paused. Okay, so also what happens is that this NV center not only has the NV of interest for us, but because this is a diamond matrix, you can see that there could be many other atoms which would be hanging around, like just the native nitrogen, which is sitting there, which did not get converted to an NV minus, but it's just the the pure nitrogen, and that has a nuclear spin, and that could behave like a toggling magnet near this NV. And then there is also this carbon 13 and other sorts of impurities. So if you look more carefully into the level structure, there is also the nitrogen nuclear spin that is hanging around, and that sort of gives you a hyperfine sort of level structure. And you can see that on either sides of the zero field line, you will have 3 lines which correspond to the 3 transitions between the 0 and the minus 1 and the 0 plus 1 levels, which are individually then split to 0, minus 1 and plus 1 because of the hyperfine. And if it were a nitrogen 15, which is the other isotope of nitrogen 14, it turns out that this nitrogen 15 is a spin half system, and therefore the hyperfine or nuclear spin half. So the hyperfine is now split into 2 only, and that has a splitting of 3.05 megahertz. Now, if we look at the Hamiltonian of this NV center, of course, there is the magnetic field term, which is just your Zeeman. And then you have the electric field, which is coupled in this format where you also can measure. So therefore, if you have some sort of an electric field, it sort of also couples to this NV center, and you can also measure the electric field. Then there is a strain, which also couples in a very similar way to the electric field. And then finally, you have temperature. Therefore, the NV center can work like, a sensor for magnetic field, electric field, strain, as well as temperature. And that really is the power of this sort of a, sensor. Now, here is another advantage of this sensor. So typically, if you have zero field, which means no field, and there is some sort of a very little strain that is present in the sample, you will see the transitions between 0, -1 and 0, +1 very ever so slightly split at zero field. Now, when you apply some magnetic field, as we discussed, there is going to be, Zeeman splitting, and you should see the lines sort of split with each on either sides, right? So this is, again, this is just, I think, the hyperfine splitting here, but then it splits into sort of the other. Now, what is interesting is that the NV center can actually happen, there are 4 different classes of NVs, because the nitrogen and the vacancy can be in either of the 4 arms of this carbon bond that is there in the diamond lattice, right? And therefore, you can have 4 different classes of NVs. And what you see here is that when you apply a magnetic field, which is, an arbitrary magnetic field with respect to the direction of this NV center, every different class of NV center sees a different projection, and therefore you see 4 different projections, essentially 4 different frequencies of this NV center. And therefore, if you can actually measure the vector components of the magnetic field here, right? And that is really the beauty of this sensor, that not only does it allow you to measure the magnetic field, which is the amplitude of the magnetic field, but also the components of the magnetic field, and hence allowing you to reconstruct back the entire magnetic field. Now, that is the power of this quantum microscope, because you are able to spatially resolve the magnetic field, but not only that, you can actually resolve the magnetic field in all 4 directions, all 3 directions, essentially. So therefore, with different, applied magnetic field, you can figure out what the entire magnetic field is going to be. Now, as I was saying, that if you have many number of sensors, as we know, typically the minimum magnetic field that is measured scales with the number of sensors and goes as square root of n. This is our typical, limit for the quantum limit for measurement of magnetic field or any sensitivity per se. But then it is also limited by the T2 star, or essentially the measurement time, which is the T2, which essentially tells you the fidelity over which you are going to really measure the quantum state, right? So it depends on the measurement time. And so better your coherence time, more you can actually measure, more number of times you can measure, and therefore better your T2, you are going to get better and better sensitivity. Again, more the number of sensors, of course, you will do more better your sensitivity will be, because it scales with square root n. But really the power of this sensor is that you can use quantum enhanced techniques and can really approach the Heisenberg limit. And that will allow us to really go to a sensitivity of n, 1 over n, where the minimum magnetic field will scale with n number of sensors, right? So denser the NV sample, the greater will be my sensitivity. But of course, we have to keep in mind that it also depends on this sort of T2, or essentially the coherence time. And there is sort of a catch-22 here, because more the number of sensors, more is going to be the interactions between them. And perhaps you may be in a regime where your T2 actually gets killed because of the fact that you have many number of sensors. Right, so there's a sort of a sweet spot that one may have to work out, and that allows you to work with the best sensitivity possible. And this really has been the prime direction in which magnetic field sensing with NV centers has been going towards, right, to optimize the sensitivity of the sensors with respect to the number of sensors as well as the coherence time. What I'm showing you here is actually the setup where we have a green laser. We modulate the laser with with an acousto-optic modulator. We have a set of lenses, and we focus it onto the sample, which is sort of sitting here on this antenna, which is used for controlling the microwave field, which is to control the level between 0 and -1 or 0 and +1, right? And here is an image where actually you can, if you look carefully, you will see that this is actually bright orange at the side of the diamond. So this is what you're seeing here is the diamond. As I'm sort of moving my mouse over this piece here, you can see this is the diamond. We focus the light in the center, and that is where the bright spot is. But what if you look very carefully, you will see that this is red in the center, in the sides, and that's essentially because of the fact that as we shine green light, and this image has been taken with a filter, so as we shine green light and we see that the red fluorescence is emitted in all 4 pi, but because diamond is a very high refractive index material, the light sort of gets trapped inside the material and comes out from the sides of the diamond, and that is what you're seeing here in this red, this sort of an orange here. And in fact, there are designs where one can actually collect the light from the red light from this side of the diamond and build sort of a side collection setup. Here we of course collect the red light on the reverse path, where we have the objective, and we collect the red light via a dichroic here and collect it and place it and sort of focus it down onto detector, photo detector, or a CCD. So what I'm showing you here is the optically detected magnetic resonance, and as expected, you can see that there are 4 different lines for optically detected magnetic resonance. And if you did sort of a frequency modulated spectrum of this, this is a zero field where you don't have any applied externally biased magnetic field. You see that this is the signature of your lock-in sort of a signal here. Now there is another, it's not linear here because there is strain, and that sort of splits this this linear nature here, and that's at zero field, and you see that little bit of electric field actually contributes to the splitting, and that is what you see here. So we can even go beyond this. What you can see is that if you sort of start applying sort of a magnetic field, you will see that it becomes nice and linear. We are just now looking at one particular line here, and you see that as you increase the microwave, which is essentially your probe frequency here, you can, or amplitude of this probe frequency, you can see that you are able to oscillate, drive the transition between 0 and -1 very effectively here, and that is what we are seeing in terms of the lock-in. So this is a typical lock-in spectra. As we scan across the different resonance frequencies, you will see that this peculiar sort of lock-in signature that we sort of see. Now, what we would like to do is now calculate what the sensitivity of our sensor is, and for that we sort of sit at one point and measure it many times, and we find out what the sensitivity of this magnetic field sensor is. And we find that in our case, our sensitivity actually is about 50 nanotesla per square root hertz for our sensors. When we are using an integrated signal, we are able to come down to a sensitivity of 50 nanotesla per square root hertz. Now, if we actually do a wide field measurement, which essentially means that we look at the magnetic signature or the ODMR from all the frequencies in various pixels, we see that we are able to again sort of image the sample with this kind of a sensor. And what you see here is something that is absolutely new and is just fresh out of the oven. Sort of here, what I'm showing you is a per pixel lock-in image here. So we are able to do per pixel lock-in measurement of this ODMR, which has not been done before, not been demonstrated before. This is the first time we are doing per pixel lock-in imaging. And what we see is that we can get a drastic improvement of the temporal imaging of this sort of a magnetic field map. Right? What you see here is if you apply a continuously modulated magnetic field with an external, say, Helmholtz coil, what we have applied here a field of about 600 nanotesla, and we are able to track very nicely the magnetic field. This in blue is the measured field with the NV center, in red is the applied field, and you can see that we are able to very nicely track the magnetic field measured for about 15 seconds here. And it can actually go beyond, we've just cropped it off here. Believe me that there is more data on the other side of 15 nanoseconds, 15 seconds actually, in fact. So in this particular setup, of course, we were doing slightly worse. We were about 215 nanotesla per square root hertz sensitivity, and therefore you see this sort of a, cruddy image here where the signal fluctuates within this 200 sort of nanotesla, but we are able to very nicely measure the 600 nanotesla field. Now, that's what we are doing right now, but what is the application that we are actually working towards? Essentially, what we want to measure are signals like this, which comes from a single neuron. So what you see here are bright spots from the single neurons. So before that, Asai, could you tell me how much time I have?
7 minutes.
7 minutes. Okay, good. So what you see here is a STED in vivo image. So essentially what is it? It's a mouse which is sitting in this STED microscope. And this is the typical way in which a slice of this brain is imaged. This is a slice of a brain with in an in vivo sort of a setup. It's a very intrusive process, as you can see, that you are essentially cutting out a part of the mice sort of mouse brain, and then you're imaging and looking at the neurons. Therefore, one would like to develop some techniques where you sort of do it outside the skull, right? You don't want to really destroy the brain and then image the neurons, right? You want to keep the person or whatever, the mammal alive, right? So that's really the goal here. And what we want to look at is the correlations between the signals that travel from one neuron to another as you sort of give some sort of stimuli. Here, the stimuli given typically to mouse are, you have a rotating disc or something like that, and the mouse sort of tries to run on this disc, and you then want to image what happens, or some sound. You sort of give some sound stimuli, and then you want to image the neurons and see which part of the brain are actually lighting up, and looking at the correlations between them. Okay, now this signal is essentially like this, where it is a null or a sort of a zero event, null or a one event, which is. So you can have like a high point and then a low point, and that sort of tells you that this is an action potential. So the information, the unit of information in a brain is an action potential as fired by a neuron. And it typically looks like this. It's about a millisecond long signal. And you can sort of think that there is a high point and a low point, and that's really the signature of this neuron. Okay, now there are methods by which people measure, or image mammalian neurons. It's a multiphoton imaging, but as you understand that whenever we use fluorescence as a technique, and if you're using multiphoton imaging, that means that light will scatter inside a tissue media, or a highly dense media which is filled with maybe water or blood, etc. and that sort of limits the depth up to which you can probe. Now, if you can have a sensor that sort of does not get limited by this, tissue nature or the blood or the dense nature of where the neurons are sitting, then you can sort of go deeper in and image deeper into the brain, right, and resolve locations and activity of single neurons much deeper into the brain. Therefore, our goal is essentially to go beyond the 1 mm limit of this, and that is really the where we think that if we can measure magnetic fields, then we should be able to actually push this limit beyond the 1 mm thickness of this brain. Okay, so what our target window of recording is essentially 400 micron by cross 300 micron depth. Now, and what we want to focus are on the frontal cortex, which is essentially plays a major role in decision making and also in higher order information processing. And these are, sorry, I don't know what happened. So these are really the sort of the focus areas where we want to image and really try to see whether we are able to image. But as you can see, like if you look at the sensitivity numbers, it really we are really quite limited, because we will not be able to go down to a depth of 1 millimeter or even beyond 1 millimeter with the kind of sensitivities we have with our sensor right now. Therefore, one of the important things that we would like to achieve is actually push the sensitivity of our magnetic field sensors to go beyond this 1 mm limit. Now, in the problem, while we know that there are challenges in terms of the sensitivity of our quantum sensor, we actually approach the problem slightly differently. We said, well, if we were given the most ideal sort of sensor, then what would we do, right? Do we have everything to actually map the neurons? Well, no, because we still don't know what the signals of a typical, a neuron really looks like. And therefore, we went back and started modeling the neuronal potentials by, thinking of them like a sort of a cable and a current carrying wire, and did a much more detailed modeling than is available in literature. And we sort of split each and every compartment of the neuron body into small little isopotential compartments and integrated out the total current. And from the current, we calculated the magnetic field by using the Biot-Savart's law in a very, very detailed way. And this allowed us to actually find something extremely interesting. What we found is that the cell body actually gives you much more, or in fact 2 orders of magnitude higher magnetic field than the axon, which is carrying the current. Okay, and this really allows us to track and formulate an inverse problem, where we say that, well, if the signal is really just located in the coming from the body of the neuron, then we can just say that we can think of it like a point source and sort of simplify the entire problem down. And that is what we did, and we found the exact magnetic field maps from the cell body and as it travels with respect to the interactions that are going on, and we formed what are the magnetic field maps as would be measured by the best possible NV sensor, right? And what you see here are actually the magnetic field maps that we can sort of measure. So we went back and said, well, now if we have the magnetic field maps, can we locate the position of this neuron, right? So for that, we sort of developed a matching pursuit based algorithm, where we said that, well, we build a dictionary of all the signatures from, single neurons sitting everywhere on a certain, volume of the brain, and find out what their magnetic field maps are going to be, and then match it with Sai.
Kasturi, yeah, just it's time. So
Yeah, I'll just wrap up very quickly. So what we did was, so I'll just skip through the the details of this, and you can find this in our recently published paper. What we did was we applied a matching pursuit algorithm, and we found out that we can very well pinpoint the location of the neurons if it were separated by about 10 micron in 2D, and in 3D we can actually go also about 10 micron with a fidelity of 84% for 2D and 70% reconstruction with 3D. Right. So, and we also said that, well, how well can we sort of differentiate this signature from two neurons that are separated as close by as 20 micron or 7, and we found that it was 20 micron for lateral separation and 7 micron for vertical separation, and we are able to very nicely reconstruct back from the magnetic field images that we would take. So that sort of gives us, the idea that, well, if we do have a sensor, then we will be able to reconstruct back the position of the neurons and be able to sort of image the neurons if we have a sensor as sensitive as the quantum diamond microscope. Now, like I said, this is not all, imaginary, because one has measured with an NV center the signature. This is a measured magnetic field map of a single neuron, but as you can see, we are talking about a complex network of neuron, and therefore our challenges are significant in terms of sensitivity, in terms of time dynamics, in terms of the wide field imaging, and therefore we are trying to sort of tackle all these sort of problems in parallelly, and trying to investigate methods by which we can image neuronal signature, in fact any temporal signature, using quantum diamond microscope. And therefore I will summarize what I have shown you and sort of told you about is a quantum diamond microscope that allows us to image magnetic field maps with very high resolution. We are yet to sort of reach the points of sensitivity where we will be able to image such neuronal signatures, but we have shown that given that we are able to measure such neuronal fields, we will be able to reconstruct back the position of the neurons. And with that, I will thank my funding agencies and the people who have been working on this, but particularly Madhur, who has been working on this particular project. And of course, there are other applications of the same sort of quantum diamond microscope that we are working on in my lab. Thank you very much. I can take questions.
Thanks a lot, Kasturi. So there is one question on Slido. Let me just make two quick announcements before I ask you the question. The first is basically that the organizers are requesting that you please turn on your video if you're comfortable with doing that, so that
yes, sure.
Feedback from not just the speaker, of course you, but also the participants, so that there's a little bit of kind of visual feedback to the speaker. The second is that there is only one question on Slido, so please use that as a method to post questions, because if you're not a panelist, then there is no way for you to actually just use the audio function to ask questions. So Kasturi, Manoj Karnekar is asking, neurons must be kept on diamond, right? Question mark. How do we do that?
Yeah, in fact, that's a very interesting question. And it turns out that neurons can be grown on top of diamond. And that's for starters. But so for the image that I showed you of the C. elegans, which is the worm was actually not kept on the diamond. It was kept very close to the diamond. It was kept about a distance of 1 mm from the diamond, with, in fact, on a glass slide, and it worked perfectly fine. They were able to image the signature of the neurons. Yeah, but if we want to actually try growing neurons on diamond, it is possible. It turns out that diamond happens to be a good substrate for growing neurons and culturing neurons, in fact.
I see. Very interesting. So Usha is asking, can you please comment on the strength and other subtle features in the avian compass patterns and that in neuron magnetic field sensing?
Sorry, can you repeat the question again, Sai?
Can you please comment on the strength and other subtle features in the avian compass patterns and that in neuron. I guess I'm not sure, but I think she's asking you to compare these two things.
Okay, so I think in the case of the neurons, we are actually trying to measure the magnetic field because the neurons are emanating the magnetic field, and we are trying to measure the magnetic field. The magnetic field that we are trying to measure is about picoteslas, or few, maybe tens of picotesla. For avian magnetic field, as you know, avian magnetic field, there exists no avian magnetic field. The birds are actually measuring the Earth's magnetic field, which is about 0.5 microtesla, I think, or 0.5 Gauss. So in that case, or 50 microtesla, sorry. So in that case, we are, I mean, the subtleties are really, as we push down, as you know, in terms of measurement of the magnetic field, the challenges are quite high, because what you see in terms of the sensitivity numbers, they are integrated sensitivity that we are talking about. So 50 picotesla reported in NV literature is actually an integrated sensitivity, which means if you use all your sensors and then you calculate your sensitivity, then you get that number. But when we want to image something and you want to really locate the position of each of these magnetic field, then you are trying to then your sensor also has to be at that level, which means that if a single neuron is actually emitting 50 picotesla, then therefore we have to have our sensor at least a picotesla, which means really the single NV sensor has to be at picotesla, and that's really the hard part of it. Yeah,
I see.
Because per pixel we don't have that sensitivity. Yeah.
Let me just take the one last question, just for time. James is asking, does the geometry of the NV centers matter? Is there an optimal geometry or arrangement of NV centers that we could?
We haven't really looked at that yet. We have always, taken a uniform distribution of NV centers. And in fact, we haven't even looked at what happens when you have, a lot of interactions. And so we just implant and we just hope that there are NV centers, and as calculate the sensitivity. We really haven't looked into the structure and all sorts of things. Yeah.
Okay, fantastic.